is the next little lecture called Basic Optics for Light Microscopy. So light behavior is different when it reaches a boundary, like a sort of a border between two media. So when light travels in a heterogeneous medium, so I remind you that our samples are very heterogeneous. We have lipids, we have proteins, all sorts of stuff. So they are made of mixed things. The light may encounter a new medium or an obstacle. So 4 things can happen then. We have reflection, refraction, diffraction, and interference. I'm sure you've heard these words, but maybe it's far away and it's a bit difficult to remember what they are, so I'm going to remind you. Let's talk about reflection first. So what happens, reflection happens at the boundary between 2 different media. So we have 2 media and we have a boundary between them. And then the light bounces off the boundary and it does not continue into the new medium. So here is the light coming in from one medium and this is the boundary. So here we have another medium, but the light does not go into this medium, it bounces off. Okay. Here you can see the representation of light as rays in the direction of light or wave fronts. So you know reflection. This is what happens on a mirror, for example. So if you have a cute cat, this is the thing that happens. The light does not pass the mirror. It just reflects and stays on the same side. So we have a change of direction and of light. And also the shape and the speed of the wavefront, so this is the wavefront, its shape and speed are not changed. The boundary is flat, so when we have a flat mirror, we get an image that looks exactly the same as the object because we have a flat mirror, right? And so then in that case, there's no deformation of the image. So this is reflection. And why is it relevant for microscopy? Well, because in the microscope we have lots of mirrors and they allow us to direct the light to the places where we want to have light going. But also something that is more relevant to us who are not inside the heart of the microscope is that when we have our sample, we also have a cover slip on the sample. And what happens is that the excitation light reflects on the glass of the cover slip. And so we will see this, you have seen this already in the video last night, is that this is the role of the dichroic mirror, is to get rid of this very strong excitation light that is reflected from the sample. So this one, reflection, is actually relevant for us. Now we have something else that can happen when light finds the boundary, it's a refraction. So refraction is what happens at the boundary between 2 different media. But this time, the light passes the boundary and it continues into the next medium. So here we have the 2 different media again. This is the boundary. Light comes in and you can see that it has a certain direction. So this is the ray optics. And we show here also the wavefront. And the light will go through into the next medium, but it is slowed down. And so the wavefront is changed. So this is what happens when you put a pencil, for example, in a glass of water. And I'm sure you've done that with your parents when you were kids. Mom, look, my pencil is broken. So there is a change of speed and there is a change of direction if the light enters with a certain angle compared to the boundary. So if the light comes in perpendicular, it just keeps going without changing its direction. But if it comes at an angle, then there's a change of direction. And the shape of the wavefront is unchanged. So it's flat here and it's also flat here. But the speed is changed and the direction is changed. Okay. And we have now something that we have heard before, is the refractive index of a medium. It's always abbreviated as n, a small n, like this. So it's the refractive index of this medium. It is the measure of how much the medium changes the speed and the direction of light compared to air or vacuum. Okay, so we can calculate this, and in here we have the reference, which is air. So n equals 1. This means the refractive index of air is 1. Glass has a high refractive index, so n equals 1.52. And then water has a refractive index sort of in between, 33. Okay, so you don't need to remember all this stuff because we're going to use them so often that you will know them by heart. And there is a little equation here that actually explains how much the light is bent. So it's just I just wanted to mention it. We're not going to do anything with this equation, but just to tell you, it's something like that. The angle depends basically on the refractive index of the media. So different wavelengths refract differently as well. So it's not just light comes in and then it changes. It depends on its wavelength. So this leads to dispersion. Dispersion is when the light is spread to a rainbow, like this. So here we have the refractive index 1. Here we have refractive index 1.52 because this is a glass prism. And when the white light comes in, so white light has many wavelengths, it comes in, it is spread because of this boundary. And then when it exits, it's spread again so we get a big rainbow. This is dispersion of white light. And why is it relevant for us? Because dispersion is used a lot in a microscope. This is the way spectral detectors work. Okay, this is the way they change the light into a rainbow so that we can acquire different colors. So that is very relevant for microscopy. So because samples are made of heterogeneous matter, they contain actually many refractive indices. So inside our sample, we cannot say we have one refractive index, we have actually many. And at each boundary, the light refracts. So that's a problem for our images, because again, the light goes like this. So when we do this, our sample preparation, when we prepare our sample, we will discuss about this later, but it is best to place your tissue section or your cells on the cover slip and not on the slide. Okay. If you cannot do that, then you should use as little mounting medium as possible. So you can see that this refraction has a consequence on the way we prepare our sample. So this is why I'm talking to you about this. It also has a consequence on the choice of objective. We need to match the refractive indices of the objective immersion medium. For example, an oil objective, the immersion medium of an oil objective is oil. So the objective immersion medium and the sample, we need to avoid their refractive indices to avoid image distortion. So we will talk more about all this, but as you can see, refraction and dispersion have a lot of consequences for us who image samples. Also, inside the microscope, light travels in air, but then every time it goes through a lens, they're made of glass, so there is refraction as well. So we can use lenses to manipulate light, and I will tell you a little bit more about this later. So reflection, refraction, and the third one was diffraction. So what is diffraction then? Again, we have a light coming in and it reaches it reaches a solid obstacle. So this is what happens at the edge of a solid obstacle in the same medium. And now we don't have a boundary between two media like we had for reflection and refraction. Now it's the same medium, but there is something that blocks the way. When the light hits the edge of this obstacle, it bends. So it continues past it, but it bends. So you can see the wavefront is changed. This is exactly why we have beautiful round beaches, because we have obstacles on the side and that bends the wavefront of the waves. So you can see here we have a flat wavefront. If I show you, you can see the lines here. So this is a flat wavefront where the waves are parallel to each other. But then when they reach this, so here far away from the pier, they continue flat, parallel. But then near the pier, they bend and they became round like this. And that carries the sound that this is the reason why many beaches are curved. Okay, so the same thing exactly happens to light. And what is the relevant for us? Every single molecule in our sample is an obstacle and it diffracts light. It depends on the size of the molecule, but all of them will diffract light. So the inner edge also of the objective is a hard boundary. So it also diffracts light. So every time the light enters the objective, we have diffraction. And image formation comes from diffraction and interference. So I will explain all this to you just in a second, but actually diffraction is super important for macroscopists. And now is the fourth phenomenon that can happen is interference. So now again, light is traveling in the same material. So we are not at the boundary between the two materials, but we have a flat wave front in here. And then we have seen that we have these islands here. So behind the obstacle, we have the light is bent, and we have this curved wavefront. But what happens in here is that we have a curved wavefront coming here and a curved wavefront coming here. And these waves, they bump into each other. And so at this area, right behind here, you see the curved wavefront here and curved here. At this area, the waves interfere with each other. Okay, and this is interference. So the wave traveling in one medium, but coming from different direction, bump into each other. And so, yes, I'm sure you remember this from your school class, that if the waves are in phase with each other, so they oscillate in the same way, they will become bigger. So this is constructive interference. And if the waves are out of phase, they will destruct each other, let's say. So it will be destructive interference, that we have a smaller wave or no wave at all in there, in these areas. So this is the amplitudes add or subtract. So it's constructive or destructive interference. So we get an interference pattern here at these places. And why is that relevant for us? If the wave is regular, so these are not regular because they are formed and influenced by the winds, but if the wave is regular and light is a very, very regular wave, the interference pattern is highly predictable. And the interference patterns carries information about the objects that the light is encountering. So the size of the object, the shape of the object, etc. So in our case, when light is bumping into everything, we get an interference pattern that tells us something about our sample. And the reason why it's very relevant for microscopy is that when we record an image, what is recorded is actually the 3D interference pattern. So I'm going to show this to you in a second. But you can see how this is very relevant for microscopy. So what we are imaging is actually a 3D interference pattern. So that means that anything we do to our sample that affects the interference pattern will also affect the image. Okay. So now we have seen what happens when light reaches a boundary, what can happen and how relevant it is for us. Now let's see, what can we do to manipulate light? So in microscopy, we use two different objects to manipulate light. We use lenses and we use irises. The lens will change the light direction, its speed and its wavefront shape. So for example, in here, I've told you before that if we have refraction and the shape of the wavefront is not changed if the boundary is flat. But when we have a lens, they are never flat, or never flat on both sides at least. So they always have one curved side. And also we have a lot of lenses in an objective. So what happens then is, because it's curved, then the wavefront is going to change shape. So here is a lens. Here we have the rays of light coming in, and then there is refraction here at the boundaries. And we can see here that we have air, and we have glass here, we have air again, so we have refraction twice. And the wave, the light comes in with a flat wavefront, it ends up being a curved wavefront. And because it's curved here, it focuses. And all these rays of light will come to one place, which is the focal point. So when we use lenses in our microscope, what we're doing is that we're changing the light direction, we change the speed, and we change the wavefront shape. And so maybe you have seen this when you were a kid as well at school, that we have here, we had a little candle, and then we have a lens, and what happens is that we show here like this, that we get an image of the candle, and the image is upside down. And then we had the second lens, and then this image is then upright. And so we have here something called the intermediate image plane. So we have an image here, And we have the final image plane, which is another image. So here we have our object, here we have an image, and here we have another image. Okay, this thing you may remember from your classes. So what happens at the sample plane is here. This is actually our object. So what do we have in our sample? We have a fluorophore. And the fluorophore is a light source. It's like a little bulb. It emits light. So it will emit light in all sorts of directions. And it emits light with a curved wavefront. So all around itself, it emits some light. So this is a little light source. And then we put a lens in front of it. That is the objective. And these are the edges of the objective. So this lens is going to change the light. And now we have a flat wavefront, okay? And then we put another lens, and then we get a curved wavefront. And what do we get here? We get an image. So this is our intermediate image plane. We have an image in there, an image of our little fluorophore. And that's the intermediate image plane. And we do the same thing again with more lights, and we end up at the final image plane where we have an image of our sample. And this is where we put the sensor. So this is where we record the image. Okay, so in microscopes, we have refraction through lenses, and the space where the images are formed are called the image planes. Okay, so this is an image plane, this is an image plane. And the rest of the space in here, everywhere, we have no image. So we have an image only at some places. Here, the light travels, but there is no image. So now I've explained to you how we can manipulate light with lenses in our microscope, but we have another object that we can use. It's irises. So this is what an iris looks like. It's a little diaphragm that you can open and close. Okay, so they are called, they can have two names, and field stops or apertures, and that depends on where they are along the light path. So I've shown this to you. We have our little fluorophore, which is tiny here, and then we get a first image, then we get a second image, and this is the image that we are recording with our sensor. Now, what happens if we put an iris right at one of the image planes? It's called a field stop if it's at an image plane. And it is visible in the image because we have an image here. And what we're doing basically when we're closing the iris is that we see black around. We block the image on the outside, so we see it. So you're going to have as one of the assignments to the coloring of the microscope, and you will have to close the field stop, and you will see this. You will see that it closes down, and the image on the outside becomes black. So it blocks the light like a curtain. This is what happens when we place it in an image plane. Oh yes, I forgot that. It only affects the edges of the image. So the center looks exactly the same here and here. But what happens if we place an iris outside of the image planes? Then it's called an aperture. And it's just a different name for it. So an aperture is outside of the image plane. And it's not visible in the image when you close it, because we don't have an image here, right? We only have image at image planes. So when we close this, we do not see the edges like this, but it does affect the whole image, as you can see here. So what happens is that it prevents some of this light from making an interference pattern here and participating in the image. So this is what happens when you place this iris here. And because we have less light coming in for the interference pattern, it affects the whole image, not just the edges, okay? So we manipulate light with lenses and with irises, and with irises, it depends where we put them. Then we have different effects. And when you will color the microscope, you will also see something like this. So it will become a little bit more in your head when you see it happen. So what is the relevance for us? Stress biologist. Well, field stops are visible in the image and they act like a curtain. So you really don't want to have a field stop closed somewhere. If at some point you go to your microscope and you see something like this, you will know that, oh, there is a field stop closed somewhere, but then you want to find it and open it. So it can be useful if you don't want this area to be illuminated. So that's going to be useful. It's also used for color illumination. And again, if you don't know what that is, I will tell you later, and you will actually have to do it on your microscope. Apertures, they are not visible in the image, but they affect the whole image. So clearly it's important for us. And it is used to adjust the contrast in transmitted light. So you will do this when you color. But the inner edge of the objective, so the objective is a big lens, and around we have an edge, right? And this acts as an aperture, okay? And that is called the numerical aperture. So we already mentioned the numerical aperture yesterday, and this is now, you know what it is. It is the inner edge of the objective, which acts as an aperture and transforms the whole image. Okay, so there is less light involved in building the interference pattern at the image plane. So we get a dimmer image if the aperture is small, and we get a lower optical resolution. So the apertures have a big influence on our images. The optical resolution of the, I remind you, what is the optical resolution? It's the resolution delivered by the objective at a given wavelength, okay? That is completely dependent on the numerical aperture of the objective. So we will talk a lot about numerical aperture of the objective in this course. The final resolution in the image can be limited by different things. It can be limited by the objective and the wavelengths, but it can also be limited by the way we record the image. And we will talk about all this, not next week, but the week after, sampling and pixel size. And it can be also affected by the contrast or the quality of the sample. So many things affect the resolution and the contrast in our images, including the aperture of the objective. So here are a few conclusions for this lecture. In microscopy, in the microscope, light encounters different media and obstacles, and that leads to reflection, refraction, diffraction, and interference. We manipulate light in a microscope with lenses that change the light direction, the speed, and the wavefront shape. And they allow us to focus the light at image planes, so that is very useful. So we can image them, then acquire the image. But we also use irises. We have, depending on where they are, they're called fill stops or apertures. And fill stops act like a curtain, but apertures are not visible in the image, but they affect the quality of the whole image. Okay, so now I'm going to have a look at what time it is. And how I'm doing with time. I think, yes, I'm doing good.